This is the website for our TCGA Next Generation heat maps. In a moment, what you'll see is an example from the breast cancer project. This is genes down the vertical axis and samples across the horizontal axis. You'll notice the classification bars at the top for various types of breast cancer. Once there is sufficient zoom, according to a Google Maps-like technology, the genes and also the sample labels show up. You'll see that one can look at the detail of this in the highly zoomed format. Fit View button at the top then gets back to the original extent of the heat map. Ribbon View will then take us to a view in which one can scan down the genes in this highly magnified format. You can see from the navigator at the upper right that we are scanning down the set of genes. Image details are just below the navigator, and classification details would show up if in fact the cursor, which is not now on the classification bar, were on the classification bar. Gene is a search term. This is ESR1. It's delineated on the heat map. And now the cursor is on the label for ESR1. We're going to gene cards and can look up any information in gene cards that we wish that's pertinent to the ESR1 gene. Going back to the heat map, the next function to be demonstrated is clicking on the dendrogram, which highlights a set of genes, a set of samples, in this case already selected for ESR1. Now going to zoom to the ESR1 across samples. Now clicking on the TCGA at the bottom of the screen. It's off screen, but that's a sample name. Here's a link to CBio. There will be reciprocal links to CBio as discussed with Chris Sander. Now back on the heat map, Fit View gets us back to the original. Now clicking on one of the clusters of genes delineates a whole set of genes, and we'll now look up pathway information for those genes. Here is a listing of the genes and then the pathways that those genes occur in, sorted in the case of the genes by frequency in the pathways and pathways frequency of the genes. Here's then clicking to get to the ideograms. These will be active and will be interactive in a couple of months. Now switch to another kind of matrix. This is the correlation coefficients is shown in a gene-by-gene matrix. Clicking on a cluster then brings up that cluster's worth of genes. Alternatively, going back to by Fit View to the original matrix, can use a drag and drop to get to another set of genes. Zooming to that region, you can see what the gene labels are showing us. Now we can look at the interactions, the first order interactions of those genes for which there is evidence. And that's the yellow dots. If we click on the green, then that brings up the literature that indicates that interaction of the genes. Here are two articles in that context. Now, it's possible to have various layers of interaction maps. In this case, we're looking at the negative log p-values to judge the robustness statistically of the patches of color that are showing up on the screen, toggling back and forth with the switch data button among the interactions, the p-values, and the original matrix, the original clustered heat map. Next can open up information on one of the pixels. In this case, it's a scattergram of the points for which we've got the correlation coefficient in the pixel on the heat map. Or we can look at the distribution of bootstrapped p-values that are shown for that pixel. Other visualizations will be there, and this will be interactive very soon as well. Now back on the original view, we can recolor on the fly. It can be a new choice of colors, or one can choose color schemes that have already been canned either by the user or a certain number that are available by default. So here is the recolored image. One can produce, and very important, a high-resolution PDF file that meets the criteria for the Nature journals and other major journals. Included with that PDF file are all of the metadata characteristics necessary to reproduce that PDF file. So 6 months later, one still knows how it was formed. And similarly, for the map itself, all of the metadata necessary for reproducibility are there, including the build history for the program and also the data that were used in forming the heat map. Again, this is very important for reproducibility. We find that it's very often the case that a considerable time later one doesn't know how these sorts of images were generated without that kind of information. Again, going back to the original color scheme which was stored, then that is the end of the video. This is a video that was put together by Bradley Broom, and hope you enjoyed it and got something out of it.